Hello everyone, welcome to my YouTube channel. Today we'll be talking about cell transport. And before I begin, I would like you to hit on the subscribe button to subscribe to this channel. You can also like and share. Alright, so before we talk about cell transport, let's just take a brief look at the key components of cell transport. Before we dive into the mechanisms of the cell transport, let's just take a quick look at the key components. So when we talk about cell transport, basically what we are trying to talk about is how substances are exchanged between the intracellular compartment and the extracellular compartment. So let's assume this to be the cell. So we are looking at how substances are exchanged from the intracellular compartment to the extracellular compartment and vice versa. And without these key components, it will be very difficult for this process to occur. Now, how the cell has been structured allows substances to move in and out of the cell, either by passive means or active means, or by other means which we will look at in the video. So first of all, let's consider the cell membrane as one of the key components that ensure cell transport. So the cell membrane has been designed in such a way that it is selectively permeable. So first of all, we can talk about what? Selective permeability. Selective permeability in the sense that it does not allow any substance that is not meant to enter into the cell, or in other words, it is highly selective in the substances that it allows to, enter the cell or leave the cell. So that is one of the cell membrane's characteristics, and this is because of its complex composition. The cell membrane consists of what? The cell membrane consists of the cholesterol itself. The cell membrane consists of the phospholipid layers. They consist of what? They also consist of the glycoproteins, and then sometimes them carrier proteins, etc. So these structures allows the cell membrane to be what? Selectively permeable. So if, for instance, the cells do not have receptors for a particular molecule or specific transport protein for a particular molecule, it will be very difficult for that particular molecule to enter. And even for non-polar substances, because of the structure of the cell membrane, non-polar substances will be very difficult to enter into the cell because of how the cell membrane has been designed. Now let's talk about the transport protein. So when we talk about transport protein, these are proteins that are responsible to move substances from the outside of the cell to the inside of the cell, and then vice versa. So these are proteins that are also found embedded within the plasma membrane. Then we talk about cholesterol. Cholesterol too has a role to play in the transport, especially when it comes to the polarity of the molecule, or whether the molecule is lipid soluble or not. So the cholesterol also ensures. And then the ATP, this adenosine triphosphate. So the ATP is actually what ensures the active means of cell transport. So we will later look at active transport. We realize that the ATP has a role to play when it comes to active transport. And then for the vesicles, it's mostly plays role in the bulk transport, where we have endocytosis. We have the pinocytosis, and then we have the exocytosis. So the vesicles plays role in the cell transport. And then for the electrochemical gradient, it also plays role, especially in the active transport. So there should always be an electrochemical balance between the intracellular compartment and the extracellular compartment. And any shift within this balance will either cause substances to move from the intracellular compartment to the extracellular, or from the extracellular to the intracellular. All right. So these are the key components. Now, aside the key components, we have, let's say, factors or concepts regarding to this cell transport that are also very important. So selective permeability, we can talk about tonicity. So under the tonicity, we might have what? Isotonic, we have the hypotonic, and then we have the hypertonic. So For the isotonic, it's when there is a balance in concentration between the extracellular compartment and then the intracellular compartment. So there is no net movement of substances either from the intracellular compartment, there's the extracellular compartment to the intracellular compartment, or vice versa. And then we have hypotonic. So for the hypotonic solution, or for the hypotonicity, this is when the concentration in the extracellular compartment is less than the concentration in the intracellular compartment. As a result, water moves from the extracellular compartment into the intracellular compartment. Then we have the hypertonic. So for the hypertonic, there's a higher concentration here in the extracellular compartment. Therefore, water rather moves from the intracellular compartment to the extracellular space, therefore shrinking the cell. So in this state, the cell shrinks. This state, the cell swells, and then in this state, the cell remains normal. So that is the tonicity. Then we have the last one. We can talk about concentration gradient. So for the concentration gradient is the difference in concentration between two areas. So the concentration gradient normally talks about the solute, but for the tonicity, it mostly talks about the solvents. That's the movement of water in and out. But for the concentration gradient, it talks about the solute. So when we take the cell, for instance, let's see, the concentration gradient will tell, let's say, the number of potassium ion we have outside the cells and the number of potassium ion we have within the cells. So this talks about the concentration gradient. So this tells us the difference in concentration. That's the concentration gradient. So probably these three facts also play huge role in cell transport. All right. Now let's see how we can classify the various types of cell or cellular transport. So cell transport can be broadly classified into, we have passive transport, we have active transport, and then we have bulk transport. Now, the active transport, the passive transport, does not use energy. So the main difference between the passive transport and the active transport is that the passive transport, no energy is required. So no energy required. So we don't make use of ATP here, but here energy is required. So ATP, but involvement. That's the main difference between the passive transport and the active transport. Now, when we take the passive transport, we will have transport such as diffusion, osmosis, and then we have facilitated diffusion. Then for the bulk transport, we have endocytosis, we have exocytosis, then we have pinocytosis. I will explain them. Then for the active transport, we have primary active transport, then we have secondary active transport. Now let's talk about diffusion. So when you take the structure of the cell membrane, there are air spaces that are found within the structure of the cell membrane that allows substances of lower molecular weight to penetrate through diffusion. So substances are able to move through the tiny spaces that are found within the cell membrane through a process we call diffusion. And then for the osmosis, the movement of water molecule from a region of lower concentration to a region of higher concentration. Yeah. So we also have spaces such as the aquaporins. So the aquaporins, for instance. So these aquaporins, they are tiny holes that are found within the cell membrane that makes the cell membrane semi-permeable, therefore allowing the movement of water molecules from a region of lower concentration, that's the extracellular compartment, into the cell, that's the region of higher concentration. So that is how the osmosis aspect of the passive transport is captured. Now, when we talk about facilitated diffusion, so the word facilitated means help. So it's a form of diffusion, but this time around, it's a protein facilitating the diffusion. And these proteins are called channel proteins, or you can call them channels. So let's use the same. So they can be in the form of these channels. and then they allow the substances to specifically pass through those channels so that they can get inside the what intracellular compartment. So that is the facilitated diffusion. Now let's talk about active transport. So for the active transport, we have primary active transport and secondary active transport. Now, for the primary active transport, it makes use of what ATP for the movement of substances from the intracellular compartment to the extracellular compartment and vice versa. And it also makes use of what protein pumps. So protein pumps. So on this side, that is where we have the sodium potassium pumps. And these sodium potassium pumps are responsible to move sodium and potassium exchangeably across the various compartments, being it intracellular compartment and an extracellular compartment. Now, what secondary active transport does is that it uses the energy produced by the ATP produced by the primary active transport to also do its transportation. It uses the energy produced by primary active transport to also do what its transportation. So that is for the secondary active transport. Now, the goal of cell transport is to move substances in and out of the cell. So all these mechanisms are purposely designed to move substances in and out of the cell. Now let's talk about the bulk transport. So in the bulk transport, we have the endocytosis and then we have the exocytosis. So normally the endocytosis is about engulfing substances into the intracellular compartment. So the cells sometimes in their way extends their cell membrane and then they engulf substances into them. So that's endocytosis. And then the exocytosis is the reverse form of the endocytosis. So when you have substance within the cell and the cell wants to expel the substance, it just creates a membrane around, and then break the membrane to release the substance. So that is exocytosis. And then pinocytosis is what is also known as cell drinking. So pinocytosis, cell drinking. So for the pinocytosis, it's more like in a fluid form. So the cells in a fluid form will capture whatever that it wants to allow to enter into the intracellular compartment. So that is it about the cell transports. In future videos, we are going to delve much deeper into them. We are going to pick passive transport. We talk about the intricacies that lies within diffusion, osmosis, and then facilitated diffusion as well. We also talk about bulk transport, talk about endocytosis, pinocytosis, exocytosis, and for the active transport, we'll talk about most of the transport proteins that are found within the active transport, including the protein pumps. So this is just an overview of cell transport. In future videos, we are going to talk much deeper into that. All right, please, I would like you to subscribe to this channel to get a lot of simplified medical concepts so that it will help you in your learning. All right, see you later. Bye. Don't forget to subscribe.